My name is Daniella, and today I will be discussing the effects of a high-fat diet on amyloid beta plaque pathology in apolipoprotein E transgenic mice. Alzheimer's disease is largely believed to be a toxic accumulation of two types of proteins. The first is amyloid beta plaques, and the second is neurofibrillary tangles. And amyloid beta plaques is what we will be focusing on in this study. And as you can see, in a normal brain, you have healthy functional neurons, but in an Alzheimer's disease brain, these neurons are cluttered by amyloid beta plaques and these neurofibrillary tangles, which then results in neurodegeneration. And researchers believe that Alzheimer's is an inflammatory disease. There's also no cure for this disease. And researchers are interested in understanding the interactions between genetic and non-genetic factors and how it influences Alzheimer's. So one genetic factor for Alzheimer's is apolipoprotein E, or ApoE. And ApoE increases one's susceptibility for Alzheimer's by approximately 50%. And ApoE is made in the brain, and it functions to recycle lipids after neurons degrade. Now, there are three types of ApoE alleles. Now, the first is called ApoE2, which is a protective allele. The second is ApoE3, which is a moderate or neutral risk factor for Alzheimer's. And the third is called ApoE4, which is a risk factor for Alzheimer's, and a person can be homozygous or heterozygous for these alleles. ApoE4 has been shown to increase amyloid beta, and this is what causes it to increase the risk for Alzheimer's. It also has been shown to regulate inflammatory processes. One non-genetic risk factor for Alzheimer's is obesity, and not only does it confer risk for Alzheimer's, but it also compromises the nervous system in general, and obesity has been linked to activation of pro-inflammatory pathways. So if we look at the big picture, we see that ApoE and obesity both work to increase inflammation, which then increases amyloid beta and causes the development of Alzheimer's. And this brings us to our study aims and research questions. And in this study, we hope to understand the independent and cooperative effects of ApoE3 and ApoE4 genetics and diet-induced obesity on amyloid beta pathology in Alzheimer's. And we expect that ApoE4 will worsen amyloid beta, and high-fat diet will further exacerbate these results. And practically, understanding the relationship between genetic factors like ApoE and non-genetic factors like obesity will help us design better treatments for inflammatory disorders like Alzheimer's. So in our experimental design, we used 3-month-old male mice. We only used males in order to avoid confounding factors due to sex differences. We had at least 6 mice per group in order to ensure reliable data, and we had a total of 8 groups. And we divided our groups into either ApoE3 or ApoE4 mice, and we further divided these mice into either control groups or experimental groups. In our control groups, mice only had alleles for ApoE3 or ApoE4, and these mice were called targeted replacement mice. And research has shown that only having the alleles for ApoE is not enough to produce amyloid beta plaque pathology. So that's why these served as our controls. So in our experimental groups, not only did the mice have ApoE3 and ApoE4 alleles, but they also had mutations for familial Alzheimer's disease. So then we used all these mice, and we further divided all 62 of these mice into either a normal fat diet, which was a 10% fat diet, or a high fat diet, which has been shown to be a 45% fat diet. So we first began by analyzing our metabolic outcomes, and we did this in order to ensure that the high-fat diet groups had poor metabolic outcomes, as expected. We measured weight and fasting blood glucose levels at 3 months of diet treatments, and at the end of diet treatments, when the mice were 6 months old, we measured glucose tolerance tests, and we analyzed glucose tolerance tests by determining the area under the curve. We then performed immunohistochemistry in order to visualize amyloid beta plaques. We did this by hemisecting the brain. We then stained the sections for an amyloid beta antibody. Just to clarify, the brain anatomy we're interested in is one, the inner cortex, which is circled in pink, and the second is the remaining part of the cortex, which is circled in red. And we assess these regions because it has been shown to be influenced by amyloid beta pathology. So to quantify the amyloid beta in our ApoE3 and ApoE4 mice, we began by taking images of either the inner cortex or the cortex. We then outlined our region of interest, so in this case we outlined the cortex. We converted this image to a black and white image, and we set the image to a constant threshold for all animal groups. and then we were able to determine the percent of amyloid beta in the region we're interested in. And by setting it to a constant threshold, we were able to identify only the most severe amyloid beta pathology. It also helped us avoid unreliable data due to unspecific staining. And I just want to also note that the same analyst was used to quantify all of the data in order to ensure inter-rater reliability, and also the analyst was unaware of the treatment conditions in order to avoid research biases. We began statistical analysis by determining descriptive statistics for body mass, fasting blood glucose levels, glucose tolerance tests, and the percentage of amyloid beta in the entorhinal cortex and in the cortex. We continued our statistical analysis by performing ordinary least squares regression analysis in order to identify the independent effects of diet and APOE genotypes, and then to understand the correlation between these two factors, we created an interaction variable. We then stratified our groups into either APOE3 or APOE4, comparing the APOE4 to the APOE3 groups, and we also compared high-fat diet to normal fat diet. Because this is an ongoing study, I wanted to point out where we're headed with this study. So in addition to completing data analysis for all of the mice, we also hope to examine inflammatory markers for obesity and APOE and the combined effects of these factors, and we also might consider investigating other markers for Alzheimer's disease pathology. Perhaps we look at neurofibrillary tangles or other stainings like thioflavin staining for amyloid beta pathology. And these experiments will also add to the reliability and the validity of our results. So thank you so much for watching, and I hope you enjoyed.